Okay, if there's no questions, we're going to begin. Let's start sharing the screen. Okay. Let's get to my syllabus. Nope, that's not what I want to know. So today is November 23rd, and this will be the only class this week because next Thursday is Thanksgiving. There will not be class on Thursday. And there is no lab today. You're supposed to be working on your unknown work. And if you identified your bacteria, you can start writing up your lab report and finalizing your unknown notes. That does need to be finalized. So make sure your unknown notes pages are finished. It's not worth a lot of points, but it is worth 5 points. Anything else I need to talk about? I got a lot of grading done. I don't remember. I think I have all the lab grades done. So all the labs are done. There are a few people who did not update lab 9, and I haven't finalized your grade, but I will do that shortly. And I've already explained to you what questions were wrong. So you can take a look at that. I think all the labs are done and all the quizzes are graded. So except for the report, let me stop sharing my screen and I'll call up my grade report, except for the infectious disease report, which I am grading. I've got, about a third of you graded. It isn't posted yet, but I have a third of you graded. And when I have everyone graded, I'll post all the grades to that. So I'm hoping to finish that this Thanksgiving holiday. So hopefully maybe about Friday or Saturday. You'll see the grade for an infectious disease project. Any questions? Like I said, I think almost everything is graded. The worksheets and the lecture in the lab are all graded, with the exception of those who in the last lab, lab 9, did not update your lab. I've already told you what the answers were that were wrong, and I'll go in there and figure out what your grade is. Generally, you lose between 0.2 and 0.4 points for each question you get wrong, so we can sort of figure out what your grade will be on that if you want to do it that way. All right, so let's continue with chapter 13, viruses, viroids, and prions. We will finish that on November 30th, Tuesday, November 30th. I will be in the lab for 15 minutes. If you have questions, I'll be there from 6:30 to 6:45. All right, so the last slide we talked about with viruses was the host range of viruses, and that's the range of cells a virus can infect. Most viruses generally infect only the cells of one or a few species. Like polio, for example, it only affects human cells. This slide is showing you the relative sizes of viruses. They are compared to a eukaryote, which we have a little bit of a red blood cell shown right here, and a bacterium shown right here. That's actually E. coli. So it's a little bit bigger than most bacteria. But viruses are small compared to a prokaryotic cell, and they're on order of about a magnitude smaller, so about 10 times smaller. Ebola is the longest virus, but it's not the virus with the biggest diameter. The bigger diameter virus would be megavirus, which is shown here. And in reality, the largest virus, megavirus or Ebola, they are approximating the size of the smallest prokaryotic cell. The viruses can be smaller than the largest viruses. And the smallest virus you know, let me see if I can blow this up a little bit. The smallest virus you know is poliovirus. That is one of the really small viruses, being only 30 nanometers, that's shown there, as composed to Ebola shown there, or megavirus shown there. And there are smaller viruses, but I doubt that any of you have heard of MS2, a bacteriophage shown right there, just a little tiny smaller than poliovirus. So poliovirus is the smallest virus that you know of at 30 nanometers in diameter. Viruses have a structure, and when we're talking about the structure, we're going to be talking about what makes up the virus and what's inside it. The virion is a term that's used for a single mature, complete infectious virus particle. So when we're talking about viruses, we could use the term virion. Virions have as their core molecule, what's inside the virus, a genome, which is nucleic acid. But unlike cells, the nucleic acid of a virus can be DNA or it can be RNA. And as you know, cells, the genome is always DNA. But in viruses, the genome can be RNA. The genome can be double-stranded or single-stranded. And double-stranded DNA is pretty common in cells. That's what most of our DNA is most of the time. But the genome of viruses can be single-stranded DNA. And let me remind you that human cells or any other cell can have single DNA. Whenever the DNA is replicating, it is single where the polymerase is actively replicating. And whenever a gene is being transcribed, the region being transcribed is also single-stranded DNA. Now, the genome of viruses can be double-stranded RNA, and that is unheard of in cells. Cells do not have double-stranded RNA. And that's where you have one strand of RNA bound with hydrogen bond. Boy, my hands aren't showing very well there. Hydrogen bonding to a second strand of RNA, very similar to the way our cells have DNA. But the genome of viruses can have double-stranded RNA. And the genome of viruses can also be single-stranded RNA. And this is common in our cells. We do have single-stranded RNA, messenger RNA. Most of tRNA and the ribosomal RNA are single-stranded RNA. When we're talking about viral genomes that are single-stranded RNA, we need to talk about whether they are positive single-strand RNA or negative single-strand RNA. The positive single-strand RNA is a strand of RNA that the ribosome can read. So if it goes UAG, didn't have that U capitalized, there we go. If it goes UAG, that is positive single strand RNA, and the ribosome can read it, and it'll start right there translating the messenger RNA. Okay, so that's the positive single strand RNA. The ribosome can read it. The negative single strand RNA is the corollary of that. So instead of A, you'd have U. Instead of U, you'd have A. And instead of G, you'd have C. And the ribosome cannot read that. Any questions about negative single strand RNA? It's just the corollary of the positive strand, or the complement. Anyways, when you're talking about single-stranded RNA molecules, you need to discuss whether they're the positive strand or the single strand, at least when we're talking about viruses. When we're talking about like messenger RNA, it's always positive single strand RNA. So we don't even call it that. We just call it messenger RNA. The nucleic acid of a virus can be linear or it can be circular. And as you recall, the genome of eukaryotic cells are linear. Their double-stranded DNA is linear. But the DNA of prokaryotic cells is circular. And viruses, I guess, they go both ways. Some viruses, like the influenza virus, are different, and they have their genome in several segments. And this shouldn't be unusual to you because our genome, human genome, has our DNA in several segments, and we call each of these segments a chromosome. Now, viral nucleic acid, we don't usually use the term chromosome to talk about the genome, because the genome of viruses don't have proteins associated with the genome, whereas in eukaryotic and prokaryotic cells, the genome does have, the DNA does have proteins in it. And the protein and the DNA is what we call a chromosome. And so the genome of viruses, we just call it the genome, or we call it DNA or RNA. Any questions about any of that? All right, so surrounding the genome, the central molecule of a virus, we have a protein coat. And this one's been cut away so you can look inside the virus. So this should be filled in. We call the protein coat around the virus the capsid. And the capsid is composed of capsomeres. Let me see if I can blow this up. So this virus, there's one capsomere and there's another capsomere. There's only one capsomere making the capsid. And there's lots of capsomeres making the capsid. The protein coat can be composed of more than one capsomeres. This one only has one that's being shown here. So they can be the same or different proteins. Whenever the virus has a tail, so this would be the head, and there's another structure, we'll show you a tail in a little bit. The tail is made up of different proteins than the head. And so there'd be different capsomeres. Some viruses, that's all there is to the virus, the central genome molecule and the capsid proteins. And if that's all the virus has, we call it a naked virus. Protein coat and then the genome. Some viruses have an envelope surrounding the protein coat. And these we call enveloped viruses. The envelope is a lipid layer, it's actually a bilayer, and it can have some proteins and carbohydrates in it. but the main molecule is phospholipid. And we'll come back to that later, probably not today. Anyways, the envelope is an external structure to some of the viruses outside the capsid. And here we're looking at a virus. Oops, too far. With an envelope. And let me see if I can blow that up. So there you see the nucleic acid, and you can sort of see the capsomere right in there. And then surrounding that is the envelope. And the envelope may be covered with glycoprotein spikes, these red or purplish spikes right here. And the spikes are important to a virus because they're the antigens, what the animal, or I guess it's only animals that have an immune system, recognize as something as foreign. And so the spikes can be recognized as foreign by the host immune system. And their spikes are important to the virus as well, because they serve as the attachment points for the virus binding to the host cell. Usually on the virus, the attachment points of the virus are the outermost molecules. And in this virus, the attachment points are on the spikes because that's the outermost molecule. And I could likely say that the antigens tend to be the outermost molecules. Now for this virus, what are the outermost molecules? Come on, somebody guess, it's not that difficult. The outermost molecules are the capsid, the protein coat. And so it's the protein coat that serves as the antigen for the host immune system. And it's also the protein coat that binds to the host cell that this virus is going to bind to, meaning the virus only infects certain cells and have to bind to certain receptors. And the molecules that bind to its host cell are the outermost molecules. In this case, the capsid coat. And in this molecule, it's the spikes that are inside the envelope, or part of the envelope. I word it that way. So you only find spikes on viruses that have envelopes. All right, let's talk a little bit about different viruses. There are three general shapes that viruses can take. There are the helical viruses, and they form a helix, which is best shown here. And somebody who did the report on Ebola virus, I've actually got that one graded. They did discuss that it's a helical virus, and we're looking at it there. And I sure don't see the helix in that. So if somebody did the report, you can explain the helix of the Ebola virus. But there's a helical virus where the virus is in a helix. And there are also polyhedral viruses. Those are viruses with many sides, and they sort of look like a dice. This has been cut out so you can see the nucleic acid inside, but that would be filled in. And there's an actual virus that's polyhedral. I can't read that. What is it? The masto virus. The third virus shape is complex viruses, and they can be any shape you can imagine. Okay, there's a complex virus, and there's a complex virus, and there's an artist's rendition of a complex virus. And there's a complex virus too. That's an envelope virus. Just trying to show you the nucleic acid inside the capsomere. And it looks like there's two segments of the genome. And then the envelope around it with spikes on the envelope. And there's an actual picture of influenza virus, which is sort of what that's supposed to be, an envelope. The complex viruses can take on any shape you can imagine. And if you look here, this one is taking on the shape of a lunar lander. Those of you who remember the lunar lander from the Apollo mission. Let's talk a little bit more about these viruses. They're called the T-even viruses. And which one is it I want to talk about? Is it bacteriophage T4? I think it's bacteriophage T4. Sorry, I'm losing my note here. The most studied virus of all viruses is bacteriophage T4. And the reason why it's the most studied virus is because it is a virus that infects E. coli, a cell that is frequently found in microbiology labs. And so because this virus infected E. coli, people started studying it. And then they said, you know, we got to really understand viruses. Let's study the bacteriophage T4. So it is the virus that has been studied the most. And for bacteria viruses, these are viruses that infect bacteria cells. We call those bacteriophages or bacteriophages. Both pronunciations are correct. And all they are viruses which infect bacteria cells. Bacteriophage T4 is a T-even virus, and they all have this type of a shape, which is the real picture of one. And this is the artist's rendition showing you that this virus has a capsid head, this portion being the head. And inside the head, you find the viral nucleic acid, which is DNA in this case, and the tail, which is this structure here. And the tail is composed of different proteins. It looks like it has a protein for, two proteins for the sheath, which is one right here, and then the outer portion of the sheath right there. And then it has the pin, so there's at least one protein for the pin and one protein for the base plate, and then a protein for the tail fibers. This whole thing is the tail, and that's the head. And viruses that infect bacteria cells, meaning the bacteriophages, tend to have both a head and a tail. And the tail is a special structure for infecting bacteria cells. When we're talking about animal viruses, they frequently do not have a tail because it's much easier to infect an animal cell than it is to infect a bacteria cell. And can anyone tell me why a bacteria cell is harder to infect than an animal cell? Nobody's going to guess? Well, animal cells only have a cell membrane, and it's much easier for a virus to infect and get through the cell membrane than it is for a bacteria virus to infect and get through the cell wall, and then get through the cell membrane. I'm not going to answer that, but I'm going to shut it off. Okay. Yeah, I don't know how to turn it off on that phone, so it's a new phone, so we're just going to leave it. All right, hopefully they'll leave a message. Any questions about that? So this tail is a special structure to get through the cell wall of a bacteria cell. Shrink that down. So when viral infection occurs in an animal, the host immune system generally will respond. So the virus stimulates the host to produce an immune response, such as the production of antibodies. The antibodies recognize and bind to viral surface proteins. These we call antigens, and they tend to be the outermost molecules of the virus. Some viral proteins can change or mutate frequently, and therefore different viral strains can infect the same individual more than once. A good example of that is influenza virus that changes faster than most other viruses. Another good example is COVID. COVID-19 is a virus that has different strains, and as you know, the different strains can infect different people, and it is possible that you can be infected more than once with COVID-19 because the different strains can infect the same person. Any question about any of that? Another reason why you might be infected more than once with COVID-19 is that your immune response will weaken with time. And so your immunity, they think, will be about a year. It might be as much as 2 years or 3 years. But after that, your immunity will weaken and you can get another COVID-19 infection, even if it's the same strain that first infected you. We'll talk a little bit more about viral infections later in this lesson, including I've got a few slides on COVID-19. All right, so when we're talking about viruses, it's important that we group them, and we call that viral taxonomy. Viruses are grouped mostly into families, genuses, species, and we can group them by their type of nucleic acid they have, whether they're a DNA virus, whether they're an RNA virus, their replication strategy. We group them by their morphology. Virus family names always end in the name viridae. So if you see something like Poxviridae, that means the pox family of viruses. The genus of a virus ends in the name virus. So the pox genus virus or pox virus. Herpes virus would be the genus. Viral species, each virus has a species. The COVID-19 is one species of the coronaviruses. A viral species is a group of viruses sharing the same genetic information and the same ecological niche, meaning they infect the same host. You'll be pleased to know that when we name viruses, we use common English names like poxvirus or poliovirus. There are no Latin names for viruses. And why this is the case is that the early virologists were all English speaking, and they started naming things with English names. And then the later people who didn't speak English, who started studying viruses, they just used the English names for things. And so it became commonly used in viral taxonomy. So you'll be pleased to know that there are no Latin names for viruses, and they all use common English names. Another thing you should know about viruses is that each species has a subspecies, and they're designated usually by a number. COVID-19, the Delta variant, is a subspecies of COVID-19. A better example is, oh gosh, let's see if I can get this right. Polio virus type 1 and type 2. Nobody calls them that because polio virus type 1 is a subspecies of the polio virus that causes the disease polio. And the other subspecies, which we'll call type 2 poliovirus, is a virus that is the live vaccine that we use to prevent people from getting polio and coming down with the disease of polio. So that tells you how different the subspecies are. They share the same genetic information. I think there's like 6 bases which are different between these two subspecies. One causes polio. The other one we use is the live polio vaccine to prevent people from getting the disease of polio. Okay, so that tells you how different the subspecies can be. Any questions about that? So the subspecies are really important, especially when we're talking about human disease. And if you don't know COVID-19, Delta variant is also very important because the different variants are very different. And they're different subspecies, at least in their infectability, how they can infect people, and COVID-19 can infect people who have gotten the the COVID-19 vaccine. Although the vaccine does give you some protection. I think if you've had the vaccine, your protection against the Delta variant is 65%, but other variants are protected by the vaccine by 95%. So what is that, a drop of 30% from the vaccine? All right, let's talk a little bit more about viral taxonomy. I sort of gave it to you before. Viruses are typically not grouped much above a family. And that's because they're just too small. Their nucleic acid is too small. And we don't know what their groupings are much above a family. Some viruses do have groupings. What's above a family? Let's see, King, Peter, son, came over from Germany. So order. Some viruses have orders, but much above an order, we don't have the viruses grouped. We group viruses into the kingdom they infect. Like there's animal viruses and plant viruses, and then they might be grouped into an order, and then they're grouped into family. And as I mentioned, herpes viridae is the family name for the herpes family. And then the genus for a genus of herpes viridae would be the simplex virus for the genus. And the common name for that, meaning there's 2 names for the simplex virus or human herpes virus. This new name, human herpes virus, is not taking hold, but it's been around now for 20 years, and the people are still calling it the simplex virus, including clinicians. The subspecies would be human herpes virus 1 or simplex virus type 1, herpes simplex type 1. And this is the virus that causes cold sores. Okay, any question about that? All right. When you're growing bacteriophages, these are viruses that infect bacteria cells in the lab. what you do is you grow up a culture of bacteria that the viruses can infect, and then you plate the bacteria on the plate so that the bacteria will grow everywhere. And then once you have the plate in the lab plated with the bacteria, before you incubate that plate, you then put on a solution containing the virus. And that would be a solution containing bacteriophages. And then you spread the solution over the plate. Wherever there's a virus, the virus will lyse that cell and then infect neighboring cells. And so you will eventually see a plaque on the plate, and the plaque is from all of the bacteria cells being lysed by the virus. So the plate has this dark color, and the white color or white off-white color is from the bacteria. And all of these plaques were where there was a virus, and then that virus replicated, lysing the bacteria cell, and then going to neighboring cells and lysing them, and eventually you see a plaque. And so this solution, I don't know, 100 to 200 viruses in this solution. And I actually did this, growing up viruses in the lab and growing plaques like this. Why this plaque is big and that plaque is small could be because initially this virus attached to the cell immediately, and so the virus replicated immediately, and therefore we got a large plaque. Whereas this plaque was small because initially the virus and the bacteria, let me see if I can put it right here, were not together. And we had to have the bacteria replicate until they came over to where the virus was. And when it came over to the virus could then infect the bacteria and then go on to infect other bacteria. And so this plaque will be started later than this plaque. And so this plaque will be larger. Any question about any of that? Okay, no questions. Let's move on to viral multiplication. The virus genome only contains a few genes, maybe a dozen, 2 dozen at most. And these include the genes like for the capsid proteins, some of the virus enzymes, generally just a few. And how does the virus multiply and replicate in the host cell? The virus hijacks the host cell. The virus infects the cell, takes over the host cell's metabolic machinery, and then uses the cell's machinery to replicate new viruses. And the virus will use the host cell ribosomes, the host cell tRNA, the host cell amino acids, the host cell's ATP, some of the host cell enzymes, and use all of those to replicate the virus. The basic mechanism for viral multiplication is similar in all viruses and all infected cells. We're going to talk about it with bacteriophages. These are the viruses that infect bacteria cells. Bacteriophages have 2 mechanisms for multiplying, meaning 2 life cycles. There's the lytic life cycle and the lysogenic life cycle. In the lytic life cycle, the host cell that is infected is lysed and killed as the virus replicates. In the lysogenic life cycle, the host cell is infected, but the host cell remains alive, and the virus and the host cell live together in symbiosis for as long as the virus remains in the lysogenic life cycle. And we'll talk more about that in a little bit. So following infection, did I go the right way? I don't think I did. I hit the wrong button, sorry. To make new bacteriophages, we need a couple of things for the virus. And what components need to be made? Well, if we have a double-stranded DNA virus, that double-stranded DNA has to be replicated. And then we need to transcribe the viral DNA to get viral messenger RNA. And then that viral messenger RNA has to be translated into the capsid proteins. And then the capsid proteins need to get together with the viral replicated DNA to make the new progeny virions. For the lytic life cycle, the lytic life cycle follows 5 stages of the life cycle: the attachment, penetration, biosynthesis, maturation, and release. The phage particles stick to the surface of the bacterial cell. They bind to specific sites called phage receptors. Next, the DNA of the phage is injected into the cell. Let me try and talk you through this because the sound isn't working. They must have updated this and this movie isn't working, at least the sound. Sorry about that. So first, the virus attaches to the host cell that it's going to infect. And we're talking about a bacteriophage, so it's going to attach to a bacteria cell. And then the tail will remove part of the cell wall. We'll look at that in a little more detail later. And then the sheath will contract, and that will put pressure on the head, and that will inject the nucleic acid from the head to be injected into the bacteria cell. Once the bacteria cell becomes infected with the virus, the first thing the virus does is it wants to take control of the cell. And an easy way to do that is to degrade the host chromosome. It's not shown here, but I'm just telling you that's what happens. And then now the virus genome is running the shots in the cell because the host cell's DNA has been cut up, destroyed. What the virus does is it then replicates its genome, which is shown here, and then it transcribes its genome to make messenger RNA, which is then in the cytoplasm read by the host cell ribosomes to make viral proteins. And we have to make the viral head and then the viral tails, which I guess are green just in this case. And they have to assemble. So the head and the tail has to come in, as well as the viral genome has to be packaged inside the head. And then the tail and the heads come together. That's assembly of the virus. And then the last thing the virus do is release. They degrade the host cell wall, which causes the cell to burst. And then that releases the new viruses, the new virions. Any question about any of that? All right, let's look at that in a little more detail. First thing that happens is the virus will infect a cell of the correct species. Remember, most viruses only infect cells of one species. So if this virus infects E. coli, because it looks sort of like T4, bacteriophage T4, then it'll infect E. coli. The virus will attach, and there will be specific binding between the attachment molecules of the virus and the host receptor cells. So there'll be a receptor right here and here. And the attachment molecules are on the ends of the tail, the tail fibers. So we attach right here, right there, and right there. And then the tail removes the cell wall right here. And then the sheath contracts. And that puts pressure when the sheath contracts on the head, much like when you contract a needle, you put pressure on the head of the needle, injecting the contents of the needle into the tissue. Well, in this case, when the virus contracts, it injects the viral nucleic acid in the head into the cell. And then the tail removes from the cell membrane, and then the cell membrane reforms. And there we see the injection of the viral genome into the cell. It's not showing you here, but the first thing the virus does is it has an enzyme for cutting up the host cell's chromosome, and that's to take control of the cell. And then the viral genome is running the cell. What the viral genome does is it replicates. So I've talked about attachment and penetration. Biosynthesis, it then replicates its viral genome. It replicates the viral proteins to make the viral capsid. And that's the TED and the proteins of the head and the tail. So that's biosynthesis. And then we have maturation, where the virus comes together. The viral nucleic acid is stuffed into the head. There's actually an enzyme for that. You don't need to know that. And then the virus self-assembles, and that is the head attaches to the tail sheath, and the tail fibers attached to the end of the tail, making the new virion. And then the last thing the virus does is it has a gene for degrading the cell wall. And when that happens, and water can move into the cell, and water will eventually burst the cell, and that will release the new virions. So that's maturation and release. Any question about any of that? Two things that happen in the viral lytic cycle is the burst time. That's the time from when the cell is first infected with the bacteriophage to the time that the cell bursts. On average, that's 20 to 40 minutes, depending on the species and the temperature. And then the burst size, that's the number of newly replicated virions that will be released from the burst cell. Depending on the species, that's usually somewhere between 50 and 200 new virions released. Any question about any of that? All right, that's the lytic life cycle, where the cell infects the host cell and then uses it to replicate and lyses the host cell. So that's the lytic life cycle. The lysogenic life cycle is a little different. Upon attachment of a bacteriophage, the DNA or RNA of the virus will infect the cell, and then two things can happen immediately. The virus, that's a lysogenic virus, can enter the lytic life cycle. And if it does enter the lytic life cycle, it follows the lytic life cycle that I just referred to you. The virus will use the host cell to replicate itself and lyse the cell, releasing the new virions. But the lysogenic virus can also enter the lysogenic life cycle. So immediately, a lysogenic virus, when it infects a host cell, it has to decide, does it want to enter the lytic life cycle or does it want to enter the lysogenic life cycle? If the virus enters the lysogenic life cycle, it will not enter the lytic life cycle. And the reason for that is there's a repressor protein of the lysogenic life cycle, which is made, and this repressor protein shuts down the genes of the lytic life cycle. And then the lysogenic life cycle continues, and the virus and the host cell will live together in harmony, you can kind of say. They'll live together for as long as the lysogenic life cycle continues. The virus remains inactive, say it's latent, for as long as the lysogenic life cycle continues. The bacteria cell is termed a lysogenic cell if it remains in a lysogenic life cycle. So let's look at that. In the lysogenic life cycle, the lysogenic virus infects the cell, it attaches and injects by penetration its host, the viral DNA or RNA into the cell. We're going to talk about DNA just because it's a little easier. And then immediately it'd have to decide, do I want to enter the lytic life cycle or do I want to enter the lysogenic life cycle? If it enters the lytic life cycle, the virus will replicate itself, both the viral DNA and the viral proteins, and it'll lyse the cell, which will then go on and infect another cell, in which case you have to decide immediately, enter the lytic life cycle or lysogenic life cycle. If the virus enters the lysogenic life cycle, the first thing it'll do is the viral DNA will enter into the host cell's chromosome. And that we call the prophage, where the viral DNA is now a part of the host chromosome. The virus only replicates when the cell divides. When the cell divides, it will replicate the host chromosome, and then one copy of the chromosome will move to this cell, the other copy will move to that cell, and both daughter cells will get the viral DNA as a prophage, and the lysogenic life cycle will continue. Let's watch a little video of that. It'll start with the lytic life cycle and then move to the lysogenic life cycle.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle. Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates, and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, so that's the lysogenic life cycle virus. It can enter the lytic life cycle, in which case it has to decide to do that right away, or it can enter the lysogenic life cycle, which it also decides right away. If it enters the lysogenic life cycle, the cell can go through many cell divisions where the virus is integrated into the host cell's chromosome that we call a prophage. And the lysogenic life cycle continues through many generations this way, living in the lysogenic life cycle. But every once in a while, the lysogenic life cycle ends for that virus. So in one cell, the lysogenic life cycle ends, and the prophage is excised from the host chromosome. And then the lytic life cycle begins. The switch from the lysogenic life cycle to the lytic life cycle is not well understood. And they've studied this now for at least 60 years, and they still do not understand exactly why the cell will leave the lysogenic life cycle and enter the lytic life cycle. And so I don't want to talk much more about that because I could. I mean, there are some things that they understand, but sometimes it leaves the lysogenic life cycle and they don't know why it enters or leaves the lysogenic life cycle. Okay? They do know why it begins the lysogenic life cycle, and that is there is a repressor protein of the lysogenic life cycle. And when that repressor protein gets made, it shuts down the genes for the lytic life cycle. All right, any questions about any of that? All right, some consequences can happen with the lysogenic life cycle. For one thing, while the virus is in the lysogenic life cycle, the host cell has a copy of the virus within it. And that will prevent other lysogenic viruses of this species from infecting this cell. So that's one consequence of the prophage in the host cell. Another consequence is that this cell has new DNA, and that DNA is from the virus, the prophage. Let's go to the next cell because I'm talking about that in the next slide. So this host cell is immune to infection from the same bacteriophage. because it's already infected and the cell is in the lysogenic life cycle. The other thing is that this cell has new DNA, and that new DNA can have new genes, and that can convert this cell from the genes of the virus. For example, cells expressing the Corynebacterium diphtherium toxin can only express the Corynebacterium toxin if Corynebacterium diphtheria, the bacteria, is infected with the lysogenic virus, because it's the lysogenic virus that produces the Corynebacterium diphtherium toxin. Any questions about that? So if you're infected by Corynebacterium diphtherium and it doesn't have the lysogenic virus in it, you cannot express, or you will not have, the Corynebacterium diphtherium toxin. And that's actually better for you because toxins are toxic to us. But if your cell, your bacteria, the Corynebacterium diphtherium, is infected with the lysogenic virus, it will produce the Corynebacterium diphtherium toxin, and then you will become more sick because your infection will be producing this toxin. So yes, your degree of illness will be dependent on whether or not the cell which has infected you is itself infected with a lysogenic virus. Any questions about that? Anyways, that's a possibility that the bacteria will express new genes that will be carried from the prophage of the virus. All right, any other questions? If not, let's turn to talking about animal viruses. We talked enough about bacteriophages, the viruses that infect bacteria cells. Now let's talk about viruses that infect animal viruses, because these are a little bit more germane to us. These will be the viruses that infect our cells and make us sick. Bacteriophages cannot infect human cells, so they cannot make us sick, at least directly. They could, if it was a bacteriophage that was a lysogenic virus, it could infect like Corynebacterium diphtherium, and then that Corynebacterium diphtherium will make you more sick because the lysogenic virus produces the toxin. So it's possible they can add to your toxicity, but bacteriophages cannot directly make you sick. I suppose it is true that if you were your normal flora were to get a bacteriophage that would kill off some of your normal flora, that could disrupt the host immune system and the normal flora balance, and that could make you sick. But generally, bacteria viruses do not infect animal cells and generally do not directly make you sick. Animal viruses do follow the basic pattern of phage multiplication, but there are several differences. Like, for example, animal viruses only have to attach to the cell membrane and get through the cell membrane. So there's a little difference in their attachment. Bacteriophages have to get through the cell wall and the cell membrane. There's a little bit of difference in the penetration. We'll talk about that. A little difference in the uncoating. The biosynthesis is pretty much the same. The nucleic acid has to be produced and the viral proteins have to be made. The maturation, there's one difference. We'll talk about that. And that is bacteriophages can only replicate in the cytoplasm of a bacteria cell, but animal cells can replicate in the cytoplasm, or they can replicate in the nucleoplasm. So we'll talk about that. And there's a little bit of difference in the release of animal viruses. So we'll talk about the differences in the release. The main difference between animal viruses and bacteria viruses is that animal viruses can replicate in the cytoplasm, but they can also replicate in the nucleoplasm. And bacteriophages can only replicate in the cytoplasm. Okay, so that's the main difference you should know about. And when we're talking about animal viruses, a rule of thumb is that a DNA virus will replicate in the nucleoplasm, and an RNA virus will replicate in the cytoplasm. Why? It's because the enzymes that the virus needs to replicate, as well as all the other things that the DNA virus needs to replicate, are already in the nucleus. So the DNA viruses replicate, the DNA animal viruses replicate in the nucleus. And then the things that an RNA virus need to replicate are in the cytoplasm. So that's why an animal RNA virus replicates in the cytoplasm. And that's a rule of thumb, okay? Trying to show you my thumb. There is one exception, and we'll talk about that later. There is one RNA virus that does not follow that rule, and that's the retroviruses. And you'll understand why when we talk about retroviruses, that they do not follow that rule of thumb. Any questions about any of that? Okay, if there's no questions, I'm going to end it here. And I will see you next Tuesday because there's no class on Thursday because of Thanksgiving. If you want to attend the lab, I will be there from 6:30 to 6:45 to answer questions. After that, I'm going to log off. All right, I'll see you later.